Imagine a security breach where the intruder doesn't force their way in, but instead convinces the guards to carry them inside. This is exactly how one of the most effective and dangerous microscopic infiltrators in nature begins its complete takeover of the body. This is Ebola virus. It is a filament-shaped virus, often appearing as a long, singular strand that can loop or twist. This simple, thread-like structure houses the genetic instructions to bypass the human immune system. These viruses originated in tropical rainforests, where they have circulated for thousands of years in a stable, long-term evolutionary relationship with their natural hosts. The primary host is the fruit bat. In bats, the virus is a silent passenger, replicating and shedding through saliva and other fluids without triggering a lethal disease response or damaging the bat's internal systems. The danger begins with a spillover event. When a single drop of infected fluid crosses into a human, the virus encounters a biological environment it isn't adapted to live in peacefully. In this new setting, it begins an aggressive campaign of replication that quickly overwhelms human physiology. Once in the bloodstream, the virus encounters macrophages. These are large security cells responsible for finding and eating foreign pathogens before they can spread. To survive, the virus exploits a process called macropinocytosis. It triggers the macrophage's outer membrane to ripple and wave, tricking the cell into thinking it is capturing a nutrient. The cell folds the virus into an internal bubble called an endosome. Inside this bubble, the macrophage attempts to destroy the intruder. It floods the space with acid and enzymes called cathepsins, which are designed to break down proteins into their base components. These host enzymes inadvertently facilitate the infection. They snip away the virus's outer layer of glycoproteins, stripping the particle down until it reveals a specific underlying molecular structure. This newly exposed protein binds directly to a receptor on the bubble's wall, known as NPC1. This specific molecular attachment triggers the wall of the bubble to dissolve, releasing the viral core into the cell's main interior. The virus has successfully bypassed the initial defenses. It releases its genetic blueprints directly into the cell's cytoplasm, heading for the machinery that controls protein production. The cell's factory lines are now under viral control. Instead of performing its normal immune functions, the macrophage begins producing thousands of viral clones, eventually exhausting its resources and bursting to release the new particles. By utilizing the body's own security cells as the means of transport and replication, the virus gains an immediate and largely unopposed advantage, spreading through the lymphatic and circulatory systems. During the dry phase, spanning roughly the first 4 days, the patient experiences a high fever and body aches. These are the result of the immune system's broad initial attempt to slow the viral spread. By day 5, the infection enters the wet phase. The virus targets endothelial cells, which form the vital structural lining of the body's blood vessels. As these lining cells die, the integrity of the blood vessels fails. Platelets, the cellular workers responsible for clotting, rush to the damaged sites, but the number of microscopic leaks quickly exhausts the body's supply. This structural failure leads to the severe external symptoms. The patient loses massive amounts of fluid through constant vomiting and diarrhea, as the circulatory system can no longer hold its contents. The actual cause of death is typically this total mechanical failure of the body's plumbing. The blood pressure drops so low that the organs can no longer receive oxygen, leading to systemic shock. Defeating this process requires immediate clinical action. In field hospitals, medical teams now utilize a combination of direct biological intervention and intensive supportive care to stop the collapse. Doctors use monoclonal antibodies, engineered proteins designed to find and neutralize the virus before it can attach to a cell. These antibodies are Y-shaped structures that bind to the virus's outer glycoproteins. They physically coat the virus, acting as a molecular shield that prevents the virus from interacting with cellular surfaces. Because the virus's surface proteins are now blocked, they can never attach to the NPC1 receptors. This halts the hijacking process entirely, preventing the virus from ever entering the cell. However, neutralizing the virus is only half the battle. To keep the patient alive while their vessels heal, doctors must provide continuous intravenous rehydration, manually replacing the liters of fluid lost during the infection. On a community level, health organizations deploy ring vaccination. They identify the contacts of an infected person and vaccinate that entire social circle, creating a human barrier that prevents the virus from finding a new host. By mapping the specific molecular interactions the virus uses to enter our cells, science can develop targeted defenses. We are learning to disrupt the infiltration at the molecular level, turning a once mysterious invasion into a manageable medical challenge.